अब हम बात करेंगे यूज़ ऑफ हीमोसाइटोमीटर फॉर सेल काउंटिंग। हीमोसाइटोमीटर ग्लास स्लाइड की तरह का एक साइटोमीटर है which was for the first time devised to count the blood cells. अगर इसको देखा जाए, this is not something a very complicated instrument. This is a glass slide which has an H-shaped structure in between. जैसे कि आप यहाँ पे देख रहे हैं, एक H-shaped structure है. इस H के ऊपर और हॉरिजॉन्टल बार के ऊपर और नीचे एक काउंटिंग चैम्बर्स बने होते हैं, जिनको भी हम अगली स्लाइड में देखते हैं. These counting chambers are the areas of interest. This is the top view of hemocytometer, and the yellow bordered area is that particular area where the cytometer is built in. This picture is the side view of the hemocytometer, which shows that the counting area is not going to be in touch with the cover slip. The cover slip is going to be supported by two mounting supports, and the space between the cover slip and the hemocytometer is going to give some space or volume for movement of the cells. When we observe the hemocytometer under microscope, it is a complex of multiple boxes. The total area of hemocytometer is 9 millimeter square. Each side is about 3 millimeter long. This 9 millimeter square area is divided into 9 boxes, and each box is having an area of 1 millimeter square. There are 4 1 millimeter square boxes which are at the corners 1, 2, 3 and 4. These areas are further divided into smaller boxes. If you can see this yellow box, the area of this yellow box is 1 over 16 millimeter square. These corner areas were meant to count the white blood cells from the blood. We have 1 millimeter square area which is right in the center of this table, which is divided into very small squares, and the area of the smallest one is going to be 1 over 256 millimeter square. When we are working with the yeast cells, we have to count the yeast cells. Our area of interest is this central region, central 1 millimeter square area. In order to count the cells, we have to take the hemocytometer like this one, cover it with the cover slip like this one, and take the sample, put minute drop at the junction of the cover slip and at the junction of the hemocytometer. Due to capillary action, the liquid will flow in and it will charge the chamber. When we observe this hemocytometer under microscope, it looks like this, and at 40x magnification it looks like this. These dots basically are the cells which we are supposed to count. As we know that the cell density, the number of cells per milliliter, is quite high in the culture media, we may need to dilute them. So we need to have dilution factor, we need to have cells counted, and the total volume of the area which we use for counting in order to find out the number of cells per milliliter, per microliter or per liter of the sample. Using this method, we can count any type of cells in per microliter or milliliter of the sample, and that number can subsequently be used to decide about the dilution of the sample.